Hello everyone, this is Hossain from the Redkin Research Center, Dhaka, Bangladesh. Today I'll be presenting exploring endogenous peptide for development of safer analgesics. Well, first I'll give you a short background information, then I'll describe the slides. We all know that analgesic drugs are two types. First one is NSAID and another group is opioids. NSAID are well known to everyone because most of them are over-the-counter drug like aspirin, diclofenac, ibuprofen. Another group is opioids, which is only used for chronic pain, say for example at the time of surgery or when we use chemotherapeutic agents. According to National Institute of Drug Abuse, more than 18,000 people died because of opioid overdose, and it's a global crisis. 2,075 million people use opioids, where the prescription rate is 43%. So now the question is, how opioid drugs works in our body? This is the presynaptic neuron, and in general there is a calcium channel, and through this channel calcium enter into the presynaptic neuron and act on the synaptic vesicle, and when calcium enter into the presynaptic neuron, it secretes the synaptic vesicle and release the neurotransmitter, which is responsible for the signal transduction. When this is the opioid receptor and opioid ligand. When opioid ligand bind to the opioid receptor, it blocks the calcium channel, and when it blocks the calcium channel, it cannot release the neurotransmitter into the synaptic gland. At the time of opioid receptor binding, it also recruits the beta-arrestin 2 to the opioid receptor, and this recruitment of beta-arrestin is responsible for the unwanted effect of the opioid drugs. So our target is to avoid the recruitment of beta-arrestin to minimize the unwanted effect of the opioids. So here are the types of opioid receptor available in our body. First of all, mu, then kappa, delta, and orphan receptors. And these are the endogenous ligands which is act on this receptor, and that helps our body pain management. And that's are the ligand we use as a drug, especially morphine, hydromorphine, meperidine, methadone are widely used as a drug. And the main drawback is these drugs grow tolerance and dependence, and the another is the withdrawal effect. We saw there are several withdrawal effects these drugs shows. So our goal is to overcome this withdrawal effect and overcome the tolerance and drug dependence. So in this study we targeted the peptides, and so using the peptides we can overcome the marketed drugs problem, especially if we use the peptides we can mimic the natural mechanism, as more than 25 peptides are already available in our body, which helps our pain management. Also, peptide acts as a bioagonism with less beta-arrestin recruitment that compared to our marketed drugs. Also, if we design a peptides for bioagonism, it lessly depresses the peripheral organ like respiratory or GI tract. So if we use the peptide as a bioagonism, we can minimize the drawbacks of our marketed drugs. As a drugs, peptides have also several benefits like it's easy to synthesis, good efficacy, high specificity, good safety, low immunogenicity, as peptides are endogenous. So it's a low immunogenic, and that is membrane permeability, high membrane permeability compared to small molecule drug. So this is the working protocol. In initial steps, we collect the peptide sequence and model by PEPFOL 3, and we prepare our protein, and finally we select the 6TTF PDB file for docking. In this study, we docked by PatchDock and FireDock, and final screening we perform molecular dynamics simulation by Desmond, and from trajectory data we analyze the principal component analysis, and to design a super peptide we also perform a structure activity relationship. In virtual screening, we used 78 peptides for initial screening through FireDock, and best 25 are screened by the HeadDock, and finally top 5 are screened by the MD simulation. Here is the final docking result. This is the opioid receptor with the transmembrane domain, and here is the binding cleft, and this is the binding pose of our best 5 peptides with control peptides DMGO. This is the simulation result. After simulation, when we analyze the trajectory, we get several data like RMSD, RG, SASA, and RMSF. From close overview, we saw that glucagon, DMGO, big endothelin showed stable RMSD profile compared to others. Here is the 4 drugs which are mostly stable over the simulation period. The another profile is RG and SASA, which is almost similar for all complexes. And at the time of RMSF, we noticed that only DMGO and crispeptin lessly disturb the protein structure as they are small in size. In principal component analysis, we use 100 nanosecond trajectory data for PC analysis, and most of the peptide grouped with APO and big endothelin, and big endothelin are shifted from this group. And for closer view, we try to explore the reason of shifting of big endothelin from the APO group, and we show that in loading plot, torsion would be the possible reason of shifting of big endothelin from this group. And here is the snapshots of after simulation. We simulate our system, membrane embedded system, and we use POPC membrane for simulation. And over the simulation period, this 2 complex was stable. That was confirmed by the final snapshot analysis. 90-degree angle view, and this is the DMGO and this is the glucagon. In more closer view, we see that there is a several residues that are bind to the DMGO and the glucagon, and most of them are binding pocket residues. We also analyze the interaction frequency and contribution of bond category. Here we see that this is the DMGO and glucagon, and these are the residues are bind over the simulation period, and the marked residues are binding pocket residues. Although DMGO have more frequency of residues, but most of them are lower frequency, which are binding pocket compared to glucagon. In bond category that contributed this binding pocket residues, at the time of DMGO, it was 61% hydrogen bond and 36% hydrophobic, and rest of them are electrostatic and others. And at the time of glucagon, it is more than 1% hydrogen bond and 28% hydrophobic, 10% electrostatic, and less than 1% are others bond type. In structure activity relationship study, here is the MLR table, and we find out the 4 major predictors. One is acidic amino acid, basic amino acid, non-polar amino acid, and net charge at pH 7. And finally, using these 4 predictors, we plot a QSR ply plot. Here is the QSR ply plot with our best 5 peptides. And in overview, you see that negative charge amino acid contributed the most, and subsequently net charge at pH 7, positive charge amino acid, and non-polar amino acid. And in MLR correlation, positive charge amino acid positively correlated, and negative charge amino acid net charge pH 7 and non-polar amino acid are negatively correlated. And we have a plan to design a super peptide using this QSR data. So in conclusion, we are trying to design a bias agonist using peptides, and we got glucagon and other 4 peptides as a emerging candidates from 27 GPCR endogenous peptides, which shows strong binding interaction with opioid receptors. After molecular dynamics, we reveal that glucagon would be a potential clinical therapeutics as it has a promising binding affinity and interaction patterns. From QSS study, we also reveal that positive charge amino acid positively correlated with the docking results. So we have a plan to design a super peptide using the QSS data. Thank you all.